Nice to see everybody here and out there in virtual land. So, why not? So, we'll begin where we left off. I know I'm presenting to everybody. So iterative. Yeah. Take a chill pill. Okay. And this is kind of where we left off. So, we'll start here and amplify. And it's about where I try to do it the same for both groups. The quick word is January and February, and even to March, we have weather. Uh, it depends. To me, I feel you always get more if we're actually here in the classroom. I realize it's not feasible for everybody. If you can get here, get here. If you can't, try to log on to listen in, as we see a lot of folks do on days like this. Because they always tell you the weather's going to be, oh my goodness, terrible. And it turns out to be not so terrible. It just would hardly have been considered a snow day, under normal circumstances. But it's changed a lot because of, and if you didn't know this, it's that because of liability. There's a great deal of concern about having people go to school when the roads potentially are a problem, or the walkways here because somebody will slip and fall, and the next thing you know, there'll be a lawsuit. So a lot of it is dictated by criteria that comes from whoever the insurance company that covers liability for this. It's a big issue. For some of you who are going into nursing or medical fields, it's a big issue. I left practice, surgical malpractice, in a field that didn't have a lot of big suits, was probably more than $20,000 a year. And that was the better part, 25 years ago. It's a long time. So there's a lot involved. Monetarily, there's a lot involved. But nevertheless, this is kind of where we were. To understand, what they try to do in any of these texts, and I've taught micro with a half dozen different texts, is about why it's valuable. Well, why it's valuable, a lot of the food supply, fermentation products, whether it's from yeast, okay, which affect bread, like sourdough breads or pizza dough for that matter, alcohol production for a variety of things, cheese production. All of these involve either bacteria or yeast, some type of microorganism, sort of adding to the party. And what's interesting is they have different microorganisms make different things. If you look, and I sort of put it out there on one of these handout things that I have on this general outline. So food production is in there, bioremediation. We use a lot of microorganisms, some of which naturally occurring, some of which we've genetically engineered to gobble up oil spills, to deal with landfills, even to deal with materials like this, cellulose, certain kinds of plastics that we ourselves have uh, uh, basically, uh, should I say, that we've created by way of microorganism, we environmentally are difficult to destroy. So we have to basically either find or develop, modify microorganisms to gobble them up. It's sort of like disposable diapers. All of that, the material from a lot of the headphones I see students, probably the material from the earbuds. Those are all materials that probably came because microorganisms could generate a certain type of organic compound. And sometimes they're difficult to get rid of environmentally. So there's a lot that's involved. And we'll get into the genetic engineering thing and the idea of gene therapy shortly. But I figured I'd just point that out to you. There's a myriad of important roles. And most of the time, microorganisms are our friends. They're helpful. They get a bad rap because there's about 2,000 of them that cause disease. And some of those diseases are life-threatening. Some of those could be bioterror weapons. There's a whole lot to that. And we're going to deal with that throughout the course of the term, I'm sure. So, wrong PowerPoint, sorry. We move on. So all of these things are part of it. Contamination. Treating microorganisms, using microorganisms to clean up debris on wounds, there's nothing new. They used to, in the old days, to pull blood out, they'd put leeches on, which actually were scientific. as they brought things to the surface. I mean, it was like, you're thinking of some medieval type of, but there was a lot of things that were done. I mean, they made infusions out of tree bark that had aspirin. That's where we got aspirin from. Moldy bread, that's where penicillin comes from. So understand that science and treatment of infectious disease perhaps has evolved from a lot of other things. And we'll get started on the evolution today. So genetic engineering and recombinant DNA. The whole idea of genetic engineering is in genetically modified organisms, and I agree that some of them are not all for good. To some extent, we tamper with food supply, and a lot of people have problems with that and understand it. And whatever your feelings are, and it's not, of course, nutrition. I mean, certainly, I have no problem. Some people prefer not to have GMO-containing materials. But for instance, we've used recombinant DNA, transferring genetic material from one organism to another to deliberately alter the DNA in either bacteria, typically E. coli, something that, particularly amenable strains of E. coli that have willingly recombinant DNA. It's willing to take DNA from something else, incorporate it, and actually change what it does or what it makes to a certain extent. And being able to utilize that, and that's what the engineering is. Most bacteria, so if we just use a generic bacteria in a rod shape, they don't have a nucleus, they have what's called a nucleoid, which is a big giant circle of superfolded DNA. Now certain ones of these readily accept DNA. Most bacteria have tiny circles of DNA called plasmids. Plasmids got about 30 to 50 gene locations, very small. And we can extract plasmids, or we can break apart other bacteria, take out their plasmids. So here is plasmid we remove, and bacteria also produce something that we're going to talk about at some length, particularly with regard to, and all this is facilitated by bacteria, because they make something called restriction enzymes. Restriction enzymes is part of their defense against invasion. We take these enzymes, and what they do is they attack DNA. So they chop DNA in a very predictable pattern. We can make DNA in the lab, and we can make it to make a particular protein because we have molecular biology. So we can assemble a particular protein, insert it into a plasmid that has been altered by a restriction enzyme being chopped apart, and effectively splice it to that plasmid, and then manipulating it in the laboratory, we can insert it into a willing bacteria and basically alter that bacteria genetically. That's genetic. And what it does, and the first product was insulin. And I told you about it the other time you were here. Basically, insulin is a well-known protein. We know the amino acid sequence. They created the DNA strand to mimic that or to produce that, inserted it into E. coli or yeast, whichever one was willing, because they're abundant, simple, easy to deal with, easy to grow organisms. And that's a big deal. I mean, you can grow E. coli in the lab. All you need is a little bit of some salt and some sugar and water, and happy days. You can grow E. coli will replicate every 20 minutes as long as it's at body temperature, continually, forever, as we used to say in my old neighborhood. Okay? And that's how insulin was made. That's what and there's other things. We make something that was used in cancer treatment called interferon, human growth hormone, which is used as well as in performance enhancing drugs, but also for people who are hereditarily or have limitations of that and are very short of stature, things along those. I mean, vaccines, hepatitis B immunizations, which most of you have had, I would dare say. Genetically modified organisms made those things. And then we do something else called gene therapy, which is different. Gene therapy means we're trying to restructure our own genes. And what we do is we take, of all things, a virus, a virus that has a particular enzyme in it that will rewrite DNA. So we create a strand of RNA because it rewrites RNA to DNA, kind of backwards. And we insert that altered RNA into the virus particle and insert that into the person. And with the cell that it will target, it will restructure and make particular proteins. The virus that we use is very famous. It's called HIV, human immunodeficiency virus. That virus that can lead to AIDS in some individuals, we take out the bad, which is its own genetic sequence, and replace it with a sequence to hopefully reformat, just like you would a hard drive to overwrite a hard drive in a computer. That's genetic engineering, which is in its infancy, everybody. But that's what it's about. And what are we using with that? Microorganisms, microorganisms, microorganisms. So there's countless examples of the uses for that. Bioremediation, like I said, oil spills, pollutants, things that take it. I mean, they've been decomposers forever. That's what they do. Bacteria and yeast, if you compost old unused stuff, my wife does it because she does a lot of organic gardening, things like that. So, we save old stuff and compost it, old grass clippings, throw it in, let it get funky and grow stuff. She grows lettuce hydroponically this time of year. Sorry. It's okay. So much salad, so little time. Not like thyme, parsley, sage, rosemary. Time. Simon and Garfunkel. See, you're not like you heard of them. God bless you. A few years ago, I said she was going to medical school. And I mentioned that, and she goes, I never heard of them. And her friend gave her one of these. They're really good. He's like, oh my God, I aged 1,000 years. Nothing aged me as much as about 4 years ago. I was teaching, not this class, but the class you just saw, the advanced anatomy and physiology. And one of the gals, about 2 weeks into the thing, came up and showed me a picture of my boys from 1985 when they were in a stroller. And it was the two boys in a double stroller because they're very close in age, my first wife and our babysitter at the time. I go, how did you get that picture? She goes, you see the babysitter? That's my mom. That was my 20-year-old student here. I aged like 1,000 years as we speak. That my 40-year-old son's babysitter, I had her daughter in your age and older daughter in my class. So here's about the 2,000 different types of pathogens, and there's a lot of infection is everywhere. Less so in this country for a couple of reasons. We are less troubled than we were 100 years ago because we have antibiotics. We're less troubled than we were 50 or 60 years ago because we have advances in medicine and affordable health care and the ability for people to get health care, even those who can't afford it. So we're medically advanced. It's not a panacea, as you will learn. We're advanced in a lot of areas and we're behind in a lot of areas. And there's a lot of different reasons behind it, and we'll delve into that during the course of the term. But this is an interesting chart. Okay, this is the United States, probably within the last 4 or 5 years. Most of the time when you see a textbook like this, the statistics are usually in that 4 to 5 year range from when they assemble it and get the data, review it, and publish it. So by the time you guys get it, 3 or 4 or 5 years old. So look here in this country, major causes of death: heart disease, cancer, accidental death. The lower respiratory, that would be emphysema and obstructive pulmonary disease, a lot of from smoking and environmental factors and work-related pollutants. Stroke, which is cardiovascular, sort of dovetails into heart disease. Alzheimer's, I don't know if that's as accurate because it's not necessarily, certainly not necessarily infectious. So there's a limited amount of Alzheimer's that we pretty well believe, a very small amount that we could pretty well prove is related to prion, similar to mad cow disease. But in a very, very limited, it was being studied here at the University of Pittsburgh. Diabetes, there's a lot of problems. I say it to every class. I'll say it to you. If you're going into a nursing or healthcare, you can't know enough about diabetes. It is this perhaps the single most common diagnosis that we see in patients. If you see a general patient population, diabetes, it leads to all of these. Heart disease, cancer, compromises your immune system, not necessarily accident, but respiratory diseases, stroke. I don't doubt that it plays a role in Alzheimer's, etc. And you can see lower kidney disease and suicide, and now influenza and pneumonia, infectious lower respiratory diseases. COVID would be in that category. Influenza, tuberculosis would be. Worldwide, different. Heart disease, stroke, cardiovascular in nature. The respiratory disease is not infectious related. Then, or now moving up from 8th place to 4th place, lower respiratory diseases a lot, and tuberculosis is a big player. And you don't even include it, it has its own separate category worldwide. Alzheimer's, other dementias, respiratory cancers, lung cancer, diabetes, injuries, diarrheal diseases. We don't suffer with those too much because of our water supply. Diarrheal diseases are more or less directly related. We have a fairly pristine water supply. Like you don't worry when you wash your hands and maybe touch something and your hands aren't completely dry and you could bring something into your mouth. Like you're going over and making something in the kitchen or making a sandwich or getting something to eat. Okay. You know, right? You don't worry when you take, now, the old style water fountains were pretty nasty. You put your mouth in, but now if you're filling up a water bottle or some type of container, you're not worried about that, are you? Probably not. Not worried at home, certainly, if you have to get a drink out of the faucet. You're not. That's because your water supply, we are very reliable. Not so in a whole lot of other areas. So diarrheal diseases are very much related to that. Just wanted to let you know. This is another category into itself, emergent and reemergent or resurgent diseases. And there's a lot, some of it is we just didn't have the technology to recognize. We knew it was a respiratory infection, we just didn't know it was a virus or a bacteria. So a lot of it has to do with, we call them emergent because we didn't see them before, but it could be advanced technology and scientific knowledge. What I think is the major cause is this mutation. Bacteria and viruses, for the most part, are inherently genetically unstable. They mutate all the time. COVID and influenza are the poster children. They mutate from year to year. That's what they do. RNA viruses, which we'll cover in this class, inherently mutate. We do not have, even when they enter our cells where we treat them, they do not have the same safeguards that we have when we process DNA. DNA viruses are a little less likely, they're not unlikely to mutate, but a little bit less so. So mutation is a really constant thing. For instance, you'll learn when we do the genetics unit that our cells probably mutated at a rate about maybe 1 out of a billion. We make a billion cells easy every day. Probably your GI tract. We get a few misfits. Our immune system gets rid of them. They undergo apoptosis. They just don't get made. When people are immunocompromised, when they smoke, when they drink, when they abuse substances, when they're not keeping themselves healthy, those mechanisms are less likely to be available, and they succumb sometimes. They get sick. Bacteria does not have the same proofreading mechanisms or genetic safeguards. We'll go over the different things that are inherently different, but mutation is part of it. Some of the mutations, unfortunately, have been the misuse of antibiotics. We do a whole chapter on antibiotics. We used to use, and this is back when I was learning about them 50 years ago. I was taking. We used a shotgun. We use powerful, we give them a big dose of something that's the most powerful antibiotic. I want to get rid of it. Absolutely wrong. You want to be very, very specific. When you overuse antibiotics, bacteria, we didn't understand it. Bacteria are living things. What do they do? What does anything do, a living thing do, when it's subjected to an adverse environment? That's the whole basis of evolution. Okay? We are able to adapt. And those items that mutate from the adverse environment that are better suited to deal with the environment survive and are more likely to reproduce, either sexually or asexually, depending on what it is. So that was the whole idea behind descent with modification, or Darwin's theory. We forgot that bacteria were living. Maybe we didn't even know they were living. So what did they do? They adapted. I mean, statistically, Staphylococcus aureus still public enemy number one when it comes to infections. 90% when penicillin was developed or identified and made in the laboratory in about 1941, 90 to 95% of all Staphylococcus aureus infections were penicillin susceptible. Now, that number is completely reversed. 90 to 95% are resistant to garden variety penicillin. And wait a minute, even the advanced penicillins it's resistant to. Now, most of the infections you're going to encounter for those who work in a hospital or even in a community, MRSA, which stands for methicillin-resistant Staphylococcus aureus. They're not just resistant to penicillin, they're resistant to penicillins that were modified to fight bacteria that were already resistant to penicillin. In other words, it's evolved in 70, 80 years a couple of times already. Been good. So these are just parts of things, other things that play a role in it. Going into areas that were not previously developed. So we start to either encounter diseases or we may or may not discover new diseases. People going out into rainforests in the Amazon, they encountered a virus that they identified as Zika, which you'll see listed here. First time we really heard about that was when the Olympics and the World Cup were in Brazil in 2014. And it's not a particularly bad respiratory virus, but it produces some significant birth defects, particularly microcephaly, small heads in babies that were born and moms who were exposed. Just for instance, just to start with something like that. If you live around here like we do, okay, I live in Cranberry Township. It has an enormous cluster of Lyme disease because they've taken, anytime there's been a wooded area, they gobble it up and build houses. And a lot of this is territory. When I walk the dog in the morning, I almost always see a deer. Okay? He also sees the deer, which is troubling sometimes because he's 75 pounds of dog. Just a big dog I can play with. No, it's a deer. Nevertheless, you get the idea. And unfortunately, a lot of those homes were built in wet areas. And whenever you have stagnant water, that's the perfect growth environment for ticks. And because of all the deer, we have the deer tick, and that's the primary host for Lyme disease. So that's one of the reasons why it is the most common, okay, what we call vector-borne infection in the United States. We're building into areas that really weren't designed for it, but for some reason have become desirable. For instance, those are the emergent diseases. Resurgent or re-emerging diseases are diseases that we got rid of. Came back. Poster child, at least in this country, are children. Tuberculosis, measles. That's right, the measles. Why is that? Well, for measles, number one, they didn't bring me this. Vaccination or vaccine laxity. People either have a distrust of vaccines or are tired of vaccines. And they really, and not all vaccines are created equal. Not all immunizations are free. I mean, the COVID vaccine, sorry, was ramped up very, very rapidly. It was never 100%, not even close to effective. But unfortunately, we had more or less what we see now, and the truth will come out. An overreaction to the COVID phenomenon, which impacted all of you dramatically, vis-a-vis your education. Okay, but nevertheless, like for instance, people don't, the MMR, the measles, mumps, and rubella vaccine, is a wonderful vaccine. Very, very low problem. Big problem with vaccines is the preservatives they use. And there's been some faulty research that people latched onto, and even though it's not 100%, although the research is faulty, the premise is right. They use some pretty nasty things to preserve vaccine. We'll get to a whole discussion. But understand that we don't, the advent of MMR, measles almost completely disappeared. Now we're seeing a comeback. I mean, within the last 10 years, an unvaccinated population was at Disneyland when a measles outbreak occurred in a group that had come from another area, and a number of people got really significantly ill. Measles isn't minor. Measles is the third largest killer after Victoria. Measles can kill. So, those are all things. As kids, we all had that stuff, and I'll tell you about it when we get into this. We call them the usual childhood diseases. We didn't even know about the identity of the viruses and what they were. Measles, mumps, germ, measles. We don't see them very often today because there's immunizations for these. And most of you had, all my kids were born in 83, 84, and 86. All had germ. That's the only disease they had. The only one of those diseases. I had all of them but the mumps, and got a mumps vaccine when about 1978, 79, because I hadn't had it yet. We just got married. We wanted to have kids. So we wanted to make sure we didn't have any problems. So vaccine laxity, number one. The one I hate to remember. We are living longer. I intentionally didn't. I had a Freudian slip and didn't mention that in the last class. So yeah. Wonder what? Travel. We travel all over the place, a whole lot. As a kid, it was a big deal if you went living in New Jersey to go to the beach. That was my big travel. And how many students you know when they go on spring break, they go, oh, we went down to Mexico. We went down there. It always bothers me. My daughter's sending me pictures. We're looking for a wedding venue, you know. We were in Cancun. What do you think, Dad? Rick, I'm here freezing my, you know what. She's sending me pictures from there. Don't have children. And my daughter's wonderful, but don't have it. So travel. And then the advent of immunological diseases and immune therapies. Because now we have a lot of medicines that are immunosuppressants that we deal with people with a variety of, you see them on TV all the time. They're advertised for ANS, they're advertised for plaque cirrhosis. You see them all the time on TV. I take them. I have an immunological arthritic condition. I take the mirror once every 2 weeks. So immunological diseases plus transplants that we do more, transfusions, all of those are things that are immunologically impactful. And tuberculosis was a condition we thought we had licked in this country. I'm not speaking worldwide. And we did it without an immunization. Okay, there is a immunization which is effective more so in children than in adults. For TB, it involves injecting a similar bacteria that's not as deadly, which comes from a similar strain that affects cows. It's got a fancy name. It's called Bacillus calumet guerin, or BCG. And it is used in a lot of countries, particularly India. You go to travel to India, whether for pleasure or for business or work or whatever, you will have to have that immunization, even as an adult, even if it's not as effective. This is required for you to get whatever, a visa or whatever, permission to travel. We never used that because it would render the TB test useless. Because from that point, you always test positive. My daughter, who I was speaking of, had that. She traveled to India at least on maybe a half dozen, maybe less, 4 to 6 times because she was an egg donor. Okay. So she, for profit, she became an egg donor and made a fair amount of money doing so. It was infertile couples. Shake it from one. Because my first wife and I were an infertile couple. We'll pay a lot of money to have children. A lot of money. So I mean, so she went through this. So I'm not terms out of school. The big problem in India, and the reason why tuberculosis is an enormous problem there, one word: overcrowding. It is the highest population of any country in the world, including China. And even though it's enormous, it's not nearly as big as China. It's about 2 billion people. Small cities have 10 million people. And it's extremely populous. That's part of the reason behind it. In this country, between TB testing, chest x-rays, and treatment when necessary, we nearly had it eradicated. With the advent of immunological diseases like HIV and the immunological treatments, we began to see something we nearly eradicated make it come back. Because HIV directly affects your immune system. So just to give you that's just some background information of these topics we're talking about. You'll learn about it. I've had not one, but 2 major ulcer surgeries. Okay, I have never tested positive for Helicobacter pylori. If they said there's an association, I'd buy it. If they said gastric cancer, I'd buy it. Ulcers by themselves are not. And every time I had an endoscopy where they insert a fiber optic tube and look around for ulcers, they do a washing or a brushing to find H. pylori. Didn't have it. So, but there is definitely a link between a herpes virus and MS, a statistical link. I don't know that it's cause and effect. Certainly aspects of heart disease. I'm not as familiar with the OCD or the obesity levels, but I certainly would not dispute that. There's been a lot of research done in those kinds of things. We do see different microbiological populations in people who are slender and people who are obese. Just for that. So all of these are part and parcel to it. And this whole idea of drug resistance, that comes from those antibiotic overusage. It's horrible. Okay. We're going to do this after. We're going to do eukarya versus prokarya. We're going to go over the cells that are like ours, even though they're microbes, and bacteria and archaea, which are considerably smaller. And again, in these PowerPoints, there's always a little bit of misinformation. It's not that they lack organelles, they lack membrane-bound organelles. Nucleus, mitochondria, endoplasmic reticulum, lysosomes, peroxisomes, those are membrane-bound organelles. Bacteria have ribosomes, they're not membrane-bound. Okay, we have free ribosomes, not membrane-bound. So those are organelles. So they're incorrect. I was going to say something a little more pejorative, but I chose not to. My dad would have said they were talking through their chapeau. Well, you and I would say they're talking out of their ass about that. See, you can't say that in class, kid. She got it. The rest of you are still asleep. It's 2:00 in the afternoon. And you've already heard that joke, so anybody I've had before, you too. Same terrible jokes, same things. They already heard about my daughter, my kids. Help me. Viruses, we'll talk about. We'll go through the whole anatomy of viruses and prions. The take-home part here, to be living, you got to have both DNA and RNA. Viruses have one or the other, never both. Prions don't have any nucleic acids. They're strictly made out of proteins. That's the big difference. And What's significant about that, particularly with viruses, is the trouble is to address viruses, we typically have to get something in there to harm them that's already in our cells. And that means it's usually toxic to our cells. So antivirals have a large subset of problems, not all of them, but many of them. And these are just some illustrations. I don't do the worms, but they're really identical sections with subtle modifications that are there. Fungi, and you do this in the lab, if you're going to deal with them, depending on who your instructor is, you classify them on how they reproduce, and these are spores, their reproductive structure. So yeah, you look at that microscopically. Vorticella is one of those things you see in pond water. It's got like a stem, and it's like movement at this sort of like cup-like structure with cilia that are how it ingests food particles. And then the other ones aren't so great here. You get much better illustrations from the lab perspective. Aha. Which brings us to the other part I really wanted to spend some time with today, and that's the historical stuff. So normally by this time, and this book has a slightly different sequence, the historical aspect is to understand that there was a lot of medical knowledge in ancient Greece and ancient Rome. And when those cultures fell, it created, with the fall particularly of ancient Rome, a lot of the instruments we use today in surgery came from instruments that were made to treat gladiators. The hypodermic needle, just one of them, for instance. So understand that in ancient Greece, they had a basic understanding. That's really where the foundations of medicine, ancient Egypt. With the fall of the Roman Empire, it created a series of about 1,000 to 1,200 years called the Dark Ages, because medical knowledge was frowned on. It was the notion, and particularly in the Western world, and I don't know enough about Eastern medicine, in the Western world was like if someone got sick, it was an act of God. If they got sick or were mentally ill, they were inhabited by the devil. There was so much misunderstanding about these things. The notion of it just wasn't there. And if you did, and if whoever the clergy person said you have to treat it this way, if you didn't treat it this way, you were in big trouble. Some pretty nasty consequences like that. So I mean, it's nothing, and you can find this in any history book in the world that are there. Their concept of disease, when someone would develop an infection or develop a growth, was called spontaneous generation. Literally means abiogenesis. Genesis, making something, bio, life, a, without. Life from lifelessness. Like you'd see in those sci-fi films, like, or witchcraft films, the sorcerer would, boom, and all of a sudden the table would start chasing you. Life, it doesn't happen. Okay? So biogenesis was the opposite of this. That's what we believe in. That particularly bacteria, which are living things, have to arise from other living things. So there was a lot, as the Renaissance arrived, we began to see the development of scientific thought, where we began to understand the whole idea of Galileo and Copernicus, and what they went through to try to convince at that point Rome and the papacy about what was effectively what was heretical thinking and what was not. We began to understand science. Da Vinci and Michelangelo, how do you think they got beautiful anatomical formations in their paintings and their sculpture? They dissected bodies, which they weren't supposed to do in secret. The original drawings, some of the anatomical drawings that you use in anatomy and physiology, were done by an artist named Vesalius in the 1500s, still being used today. They got those from dead bodies. Be clear. So as we began to see a change in scientific thinking, the major challenge was to dispute abiogenesis and spontaneous generation. And surely the most important figure in all of this Eventually, it was Pasteur. And we'll spend most of our time talking about, and it's going to come here, but we're going to skip it and go back historically. So Pasteur and the swan neck flask experiment probably is the most significant, although flawed, experiment that proved or disputed or debunked spontaneous generation or abiogenesis. Effectively, it's called germ theory. Tiny microscopic particles make diseases, and we know this. Okay, and we'll go into it because I have better illustrations in another textbook. So I'll take you over to here, and we'll give you two of these in the time we have left. The first one is Redi. Redi was an Italian biologist in the 1600s, around 1660. And he made, and this is all basic scientific theory. You make an observation, get an idea what you can prove with it, and create an experiment to prove it. He saw that when meat was left out, as soon as flies landed on it, shortly thereafter, maggots started to grow. And he reasoned that the flies were laying eggs, and the maggots weren't something that magically created out of dead meat. Okay, but they came from eggs that were hatching, and the meat was basically a food source. Okay, he was right. How do I prove it? Well, to them, abiogenesis meant there was some life force in the air that could magically create things. Well, I'll take 3 pieces of meat, identical meat. I'll put one in a glass petri dish, just like the ones you use in the lab, and cover it with a glass top so no air can get to it. And it didn't grow maggots, as you might guess. Had another one he left open to the air. As soon as the flies were in town, maggots. He covered another one with cheesecloth, fine mesh gauze, reasoning that that would allow air to get to it, but would not allow the eggs of the flies to get in contact with the meat. And indeed, he was right. It didn't develop maggots. Everybody said, "Oh," and everybody debunked it because in science, when you validate something, somebody else does the same experiment and should get the same results. Well, they didn't, mostly because they didn't use the exact same cheesecloth. That's what the fine mesh goes with cheesecloth. He was in Italy, and if you ever made cheese on your own, that's how you make cheese. You remember the curds and whey thing? The curds, you put them in cheesecloth. Typically, you hang them, let the water come out, or put a weight on them, and you put them in a form. That's where you get the term formaggio from cheese. I've done that. I've made paneer, which is Indian farmer's cheese. You got to cook more. You'll get there. Nevertheless, so if you didn't use the correct cheesecloth, if the holes were too big, eggs are going to get through. Experiment this wrong. But he was right in his basis. Okay. After that, and before, now Pasteur was roughly about 1865, 1868, just after our Civil War. Okay. There were a couple of others. The one we'll talk about most importantly in the time we have left is Edward Jenner. Edward Jenner and his experiments, at great risk to himself, were able to eventually eradicate one of the greatest killers of all time. The greatest killer by infectious disease, I think, in the history of the world, in one particular problem, is called the Spanish flu that occurred between 1918 and 1919. It was a variation of a bird flu, which we'll talk about because it had bird DNA or RNA rather in that particular virus. Not unlike some of these COVID strains, having multiple antigens called a novel NOVEL or new virus, something that our immune systems respond inappropriately to it, very aggressively to. The other viral disease, which is the ultimate bioweapon, smallpox. Jenner in 1791, long before Pasteur, long before anything historically, he basically made an observation, scientific method. Smallpox was all over England at the time, and he saw that milkmaids who got things on their fans called cowpox didn't get smallpox. And he reasoned that there's something in the cowpox. Pox means a pustule on the skin, was keeping the smallpox away. So he figured if I take out some of the fluid from pox, somehow refine it and inject it into somebody and expose them to smallpox, they won't get sick. And it was interesting. Why is it a great personal risk? If you give somebody an injection from a pustule from a cow in somebody's finger and send them into a room filled with people with smallpox and somebody dies, you're going to be convicted of murder and or whatever. However, whatever they did back in 1791 England. It wouldn't be pleasant. There's still probably George III, the crazy guy with the birthday parties, Revolutionary War. Remember him? Did you take history in high school?
It's okay.
They're killing me. George III, Crazy George, Queen Victoria's great-grandfather. It's a little, we say in Maya, what's a little Meshuggah. A little bit mixed up. Nevertheless, he was right. And he created the first real immunization. Okay? What do we call immunizations? Vaccines. Why do we call them vaccines? What's the word root in Latin for cow? Vacca. From cowpox. So that was how we derived vaccination. And ladies and gentlemen, that was 1791. I think the vaccine was historically 1793. By 1977 is the last recorded case in Somalia of smallpox, which has been effectively eradicated, to our knowledge. We still keep smallpox vaccine. The government does, just in case. I'm sure there's smallpox cultures, just in case. That might be used for biologic warfare. Don't think for a minute that we don't think about that stuff. If the bad guys do it, you have to be ready to respond. And typically and if you did not know, we all of your grandparents and great grandparents, myself included, got immunized for smallpox. It was a pretty nasty in the old days. Like my mom and I remember grandmothers having these big looking, weird looking things on their skin that were really sort of the fallout from the way they used to do the vaccines. They would put them on. It was pretty noxious and create a pretty big sore. It was very, very effective. And that was the saga of Jenner. We will deal starting Monday with Semmelweis and get to Lister and all these other guys and Koch and go from there. Thank you very much. Have a great weekend. Oh,